Parkinson's syndrome. A deep dive into pathophysiology. Introduction to Parkinson's syndrome. Parkinson's syndrome, or Parkinsonism, is a neurodegenerative condition characterized by motor and non-motor symptoms. It results primarily from the progressive loss of dopaminergic neurons in the substantia nigra pars compacta and dysfunction in related neural circuits. Let's explore its mechanisms and the intricate pathophysiology underlying this disorder. The role of dopaminergic neurons. Dopaminergic neurons are primarily located in the substantia nigra, a structure in the midbrain, and project to the striatum, part of the basal ganglia. These neurons release dopamine, a critical neurotransmitter for regulating motor control, mood, and several autonomic processes. Substantia nigra pars compacta produces dopamine, which modulates activity in the striatum. Striatum integrates dopaminergic input to regulate direct, D1 receptor-mediated, and indirect, D2 receptor-mediated, motor pathways. In Parkinson's syndrome, up to 70–80% of dopaminergic neurons in the substantia nigra are lost before motor symptoms appear. Pathophysiology of Parkinson's syndrome. Neuron degeneration in the substantia nigra pars compacta. Oxidative stress. Excessive production of reactive oxygen species, ROS, during dopamine metabolism damages neurons. Mitochondrial dysfunction. Reduced activity of complex I in the electron transport chain impairs energy production. Protein aggregation. Misfolded alpha-synuclein proteins aggregate into Lewy bodies, which disrupt neuronal function and promote apoptosis. Neuroinflammation. Activation of microglia and release of pro-inflammatory cytokines exacerbate neuronal damage. Imbalance in the basal ganglia circuitry. The basal ganglia govern motor control through two primary pathways: the direct pathway, which facilitates movement, and the indirect pathway, which inhibits movement. Loss of dopaminergic input reduces activity in the direct pathway, leading to impaired initiation of voluntary movements. Increased activity in the indirect pathway results in excessive inhibition of motor programs, manifesting as bradykinesia and rigidity. Neurotransmitter dysregulation. Dopamine deficiency. Primary driver of motor symptoms such as resting tremor, bradykinesia, rigidity, and postural instability. Acetylcholine imbalance. Overactivity of cholinergic neurons in the striatum contributes to tremors. Other neurotransmitters, serotonin and norepinephrine systems are also affected, explaining non-motor symptoms like depression, sleep disturbances, and autonomic dysfunction. Molecular mechanisms of neurodegeneration. Alpha-synuclein and Lewy bodies. Alpha-synuclein, a presynaptic protein, aggregates into insoluble fibrils within neurons. Lewy bodies, the hallmark of Parkinson's pathology, are primarily composed of alpha-synuclein and ubiquitin. These aggregates interfere with synaptic transmission, axonal transport, and cellular homeostasis. Genetic factors. Mutations in genes like SNCA, alpha-synuclein, LRRK2, leucine-rich repeat kinase 2, and Parkin are implicated in familial Parkinson's cases. PINK1 and DJ1 mutations disrupt mitochondrial quality control, promoting oxidative damage. Environmental triggers. Exposure to toxins like MPTP, pesticides, and heavy metals can induce Parkinsonism by selectively damaging dopaminergic neurons. Non-motor features of Parkinson's syndrome. Beyond motor dysfunction, Parkinson's syndrome includes a range of non-motor symptoms due to widespread neuronal loss and neurotransmitter imbalances. Cognitive impairment. Loss of dopaminergic input to the prefrontal cortex contributes to executive dysfunction. Progression can lead to Parkinson's disease. Dementia, PDD, associated with Lewy body pathology in cortical areas. Autonomic dysfunction. Includes orthostatic hypotension, constipation, urinary incontinence, and sexual dysfunction, caused by degeneration of the autonomic nervous system. Sleep disorders. REM sleep behavior disorder, RBD, is an early sign, reflecting neurodegeneration in brainstem regions. Neuropsychiatric symptoms. Depression and anxiety are common due to serotonin and norepinephrine dysregulation. The Unified Parkinson's Disease Rating Scale, UPDRS. Doctors use the UPDRS to assess the severity of Parkinson's symptoms. It evaluates mentation, behavior, and mood, cognitive and psychiatric symptoms. Activities of daily living, ADLs, functional independence. Motor examination, tremors, rigidity, bradykinesia, and postural instability. Treatment complications, dyskinesias and motor fluctuations from long-term therapy. Treatment approaches. While there is no cure for Parkinson's syndrome, treatment aims to manage symptoms and improve quality of life. Pharmacological treatments. Levodopa, the gold standard treatment, converted to dopamine in the brain. Often combined with carbidopa to prevent peripheral metabolism. Dopamine agonists mimic dopamine's effects on D1, D2 receptors. Examples: pramipexole, ropinirole. MAO-B inhibitors reduce dopamine breakdown, e.g., selegiline, rasagiline. Anticholinergics address tremors by countering acetylcholine overactivity. Surgical options: deep brain stimulation, DBS, electrodes implanted in the subthalamic nucleus, STN, or globus pallidus interna, GPI, modulate abnormal basal ganglia activity. Lesioning procedures, rarely used but effective in reducing tremors. Emerging therapies: gene therapies targeting alpha-synuclein aggregation. Stem cell-based approaches to restore dopaminergic neurons. Neuroprotective agents targeting mitochondrial dysfunction and oxidative stress. Conclusion. Hope in understanding. Parkinson's syndrome is a complex neurodegenerative condition affecting millions worldwide. Understanding its pathophysiology not only helps us manage symptoms but also fuels the pursuit of curative therapies. By combining advanced research with compassionate care, we can continue to improve the lives of those affected.